right, well, thank you very much. That's your applause. So thank you very much, Thomas, for inviting me, and thank you also to Vina for enabling this this visit and for the kind introductory words. So it's a pleasure to be here and to be presenting some work that we've done over the last few years on cognition and brain structure and function in the schizophrenia spectrum. So most of the studies, in fact, that I'll show today are not done on patients with a diagnosis of schizophrenia, but rather on people who are in what we consider the spectrum of schizophrenia. And I'll try and describe first very carefully what I mean by that. So what is this phenotype that we call schizotypy, and how does it relate to schizophrenia? So that'll be the first part of the talk, defining and describing what schizotypy is and how it relates to schizophrenia, how we can differentiate it from schizophrenia. And then in the next big part of the talk, I'll show you some evidence on the relationship between the two. And I'll focus on our brain structural, brain functional, and some aspect of cognition studies in schizotypy. And then in the last part of the talk, which will be the shortest, so by the time we're there, don't worry, we've nearly made it, I'll sort of try and turn things around and say, okay, now we've learned a lot about schizotypy. How can we use this? And you're clinicians, most of you are clinicians or aspiring clinicians, so we and others try to make a contribution, not just in terms of basic science, how does the brain work or how does it maybe malfunction in psychiatric conditions, but also what can we do with the knowledge that we've gained, and how can we use this in order to maybe support drug development, as an example. Okay? So first of all, what is schizotypy? It's basically a set or a cluster of personality traits. Okay? So it's really important to bear this in mind, that almost all of the studies I'm going to show you are done in healthy individuals. And by healthy, I mean, and that's a very big question, of course, in medicine and medical ethics, what is healthy, what is unhealthy, who should be treated, who should not be treated. But by healthy, I mean people generally without a DSM diagnosis. Okay, some of these may have a DSM, what used to be access to diagnosis of schizotypal personality disorder, but really I mean a personality, a set of personality traits, and we could measure this in all of us, and we'll all differ, and I mean the people with high scores in these traits. Okay? So what we measure with a questionnaire, and I'll show you some questionnaire items in a few minutes, are thoughts, attitudes, feelings, behaviors, experiences, emotions that resemble the symptoms of schizophrenia in a weaker, in a subclinical form. And when you look at these symptoms, at what's called the phenotype, at the observable, reported behaviors, experiences, feelings of individuals, and you do some statistical analysis on them, factor analysis, for example, then you realize, well, these cluster together in 3 dimensions: the cognitive, perceptual, interpersonal, and disorganized dimensions. And these look a little bit like the symptom dimensions of schizophrenia that Little and others have identified, of positive, hallucinatory, delusional symptoms, thought disorder, and negative symptoms. So cognitive perceptual means things like magical thinking, unusual perceptual experiences. Note, I'm not really saying hallucinations. There may be people who have the odd hallucinations, but I mean just altered perceptual experiences. Ideas of reference rather than delusions. So that's a cognitive perceptual, positive cluster, if you like. The negative is the interpersonal, so that means deficits, difficulties in relating to other people, lack of interest, no close friends, reduced affect, social anxiety. And then there's what's called the disorganized dimension. That really is thought disorder, again at a subtle subclinical level. So anything from intruding thoughts to odd speech, odd and eccentric behavior. There may be other dimensions. Statistical analysis don't always agree. Factor analysis is a volatile tool, and what you get out depends on what you put in. But by and large, there's agreement that there are these 3 dimensions, and they resemble a little bit the symptoms of schizophrenia, but they're found in healthy individuals. And that's a pretty invariant factor structure regarding a number of socio-demographic variables, in case you're wondering, well, does this depend from one location to another? Not really. So how do we measure this? With questionnaires. There are a number of questionnaires. They're literally self-report questionnaires. People go through these questions. They answer either yes or no, or I agree, I don't agree, or maybe varied response formats. Now, what does someone look like who scores very highly on these questionnaire measures? So a typical schizotype, if you like. And this is a quote from a book by Mark Lenzenweger, who's one of the protagonists really in schizotypy research. And he describes the model schizotype as follows. The model schizotype is someone who has relatively impoverished interpersonal relations. So that's the interpersonal domain, negative symptoms, if you like, often having few friends. Not because they don't succeed, but they don't seem to be interested in having friends. The manner in which the schizotype speaks and uses language can be rather unusual. That's the disorganized dimension. How do we know if someone has thought disorder? Where we observe, we listen to what they say. That's a pretty good indication of what the thinking processes are like. The schizotype may have momentary oddities in perception. That's the cognitive perceptual hallucinatory-like domain. For example, seeing objects or body parts briefly change their shape or size. And in fact, it was Jeffrey Gray who taught me this on said master's program at the IOP. And I can't remember which book he took the quote from, but it was a book by Albert Camus, I think. And he said, there's a perfect description of this. There's this guy who's looking at his hand and says, it looks different. It's just changed its appearance. And maybe you know this from, maybe from feverish states, but there are these, I'm not talking about proper hallucination, but about changes in actual perception. So that's what's meant by this here. Then relatively neutral events may have special significance. There may be messages on TV, perhaps. People are talking about me. So this is the delusional aspect of cognitive perceptual symptoms. The person may think that he can make things happen as if by magical ability. Emotions and affect expression are rather atypical. A schizotype may display little, if any, affect, may claim to feel little to no emotion, report feeling highly anxious around people, and may be suspicious. Okay? So that would be a model schizotype. And you will recognize quite a few, if not all of these traits from patients and maybe from friends and colleagues, and I don't know. But I mean, it's really it's meant to be a subclinical expression of the symptom structure of schizophrenia. That is the point. So how do we measure this? With questionnaires. Here's a questionnaire that is widely used, the SPQ. It's one that yields 3 dimensions. Items from the cognitive perceptual dimension are you sometimes sure that other people can tell what you're thinking? Okay, so there's a sometimes in there. Yeah, it's not, do you actually believe this, but sometimes maybe. And then people would say, yeah, not always, but yeah, sometimes I actually agree. Have you ever noticed a common event or object that seemed to be a special sign for you? Okay, so you have to word this very subtly. Interpersonal, do you feel that you're unable to get close to people? Or I tend to keep in the background on social occasions. Disorganized is, I sometimes use words in unusual ways. And people can reflect on what they say and how people respond to them, and they might say, yeah, actually sometimes I invent new words or I make links between words that other people haven't made, and then people look at me like this, and so it's obviously maybe a little odd what I say. I find it hard to communicate clearly what I want to say to people. That's an interesting variant of it. So here someone is trying to get a point across, and their thoughts are sort of messing things up and they can't get the point across. So this is what it looks like. This is what schizotypy looks like. And now the question arises, how does this relate to schizophrenia? And maybe let's start with schizophrenia. Is schizophrenia a binary phenotype? Is it something you have or you don't have with sudden onset? Like a broken leg, that's the best example. It breaks a second before it wasn't broken, now it's broken. It's either broken or it's not broken. So that is a very clear, very simple binary phenotype. Is schizophrenia like that? Well, probably not. And there are certainly not. And there are a number of hypotheses that have been generated to explain various aspects of continuity around schizophrenia. And it's really about continuity or continua between health and disease. And this applies not only to schizophrenia, but to any aspect of disease, physical disease included. So there are different forms of continua that have been examined. I'll focus really on the last one of these, the phenotypic continuity, but there's a genetic continuum as well by studying relatives, something we did excessively in studies in London. By studying relatives of schizophrenia patients, we try to get at this genetic continuity. There's a developmental or temporal continuity, of course, including a premorbid and prodromal phase before illness onset. So it's not a sudden onset disease. And then there's a phenotypic, or at the population level, continuity, and this is really what I'm talking about. Yes, I'm glad you're asking, because I actually wanted to say, if you have any questions, interrupt and do ask.
On the genetic part, what are the odds of passing that on to your children?
Well, I can't really answer it like that, but genetics is a pretty strong, I think it's the strongest risk factor for developing the illness, and the prevalence of schizophrenia in the relatives is about 14 to 15% versus about 1% in the general population. So just to give you an idea of the sort of numbers.
Okay, thank you.
And in terms of the genetics, I won't say any more about it, but schizophrenia seems to be a highly polygenic disorder, so there doesn't seem to be one gene or even a major gene or even just a handful of genes. They're likely to be hundreds of polymorphisms that each contribute just a little bit of risk to developing the illness, and then environmental factors come into play and may buffer some of the gene's effects, interact with gene effects, and so on. Okay? Yes, and please do interrupt and ask. I mean, it's meant to be partly educational, I guess, so there's no point in me carrying on talking if you haven't, if I've lost you, so do interrupt. So let's look at the continuum again at these different levels of continuum. And here's a very interesting study concerning the phenotypic continuum. So the question is, can we find schizophrenia-like traits or symptoms in the general population? And there's a study, and that's not a typo. It really is from the 19th century, a large-scale survey. I mean, I wish we had these sort of sample sizes in the 21st century, but this is a survey done through the Psychic Society in Victorian Britain. And the ideas we have of Victorian Britain and Victorian England is that people are probably quite uptight and they might not admit to odd things. But look at this. Out of these 17,000 adults, approximately 10% reported that they had at least one hallucination in their lifetime. That's an amazing figure. But it was replicated with very similar numbers by Jim Van Ars and colleagues who did similar population-based surveys by telephone in the 20th century in the Netherlands, and they found very similar estimates. So there are a lot of people, 10% is a lot of people, who have had a psychotic symptom at some point in their lives. That's a phenotypic continuity. The genetic continuity, well, again, there are old descriptions by Kraepelin and Bleuler and others who observed behaviors and symptom-like expressions in the relatives of the patients they were seeing. And this would suggest that it's a familial but possibly genetic effect, so that even if those relatives didn't develop the illness, they inherited some aspect, some subclinical form of it, perhaps. And more recent evidence supports this at the level of cognition and brain structure. Again, some of the studies we did in London would support this as well. And then the psychology has made a big contribution to this debate. Differential psychology, that's the psychology of individual differences. How do we differ in personality? Why do we differ? What are the factors that make us differ? And individual differences, psychology, as well as clinical psychology, has been arguing for quite a long time that there is this continuity, these between health and disease. And some of the names that will keep coming up in this sort of literature are Paul Meehl from the US. He coined the term schizotaxia. Hans Ising from the Institute of Psychiatry, and Gordon Claridge also from the UK, from Oxford. They've been arguing for a fully dimensional approach for the continuum between schizophrenia and non-schizophrenia or health. Okay, so the sort of questions we've been addressing are, given this similarity just in terms of the way this looks, by asking people, how do you feel, what do you think, how do you behave, we see the similarity between schizophrenia and schizotypy. Now, where does this come from? If this is true, can we also find similarity? Is there a continuum also at the level of the brain? And we can measure brain structure, we can measure brain function, cognition, oculomotor control, and we can measure the drug response. We can measure everything that has been measured in schizophrenia and try and replicate or see whether we find traces of that in high schizotypy. And we and others have been doing that, and this is relevant for a number of reasons. The primary reason really is that eventually this work will tell us something about the etiology of schizophrenia. What are the factors that lead to brain structural, brain functional patterns that are associated with high schizotype, and what differentiates the high schizotypes from people who get a full-blown diagnosis of schizophrenia? How come as a proportion of individuals can have these symptoms or these unusual traits and not develop schizophrenia? So what positively discriminates them from schizophrenia patients? I think that's the most important reason for studying schizotypy. But then it's important to understand schizotypy in its own right, because people with high scores on these schizotypy scales also engage in a lot of unhealthy behaviors, like taking drugs. They don't achieve as much as people with normal or lower levels of schizotypy. They even have fewer friends on Facebook, and so on and so forth. So they're generally not as well. They have affective disturbances. They might not report to a GP, or they might not come to your clinic for being schizotypal, but they might come because they have an anxiety disorder or an affective disorder. And a final possible reason is the prediction of conversion to schizophrenia, but that is a less likely thing to happen, because most, in terms of what we know from longitudinal studies, most people with high schizotypy scores remain like that. So that again takes me back to the first point. Well, how is that possible? What differentiates them from people who have schizophrenia? And then we can apply this knowledge, and that's like an excursion or sort of an outlook I'll give in the last part of the talk. Can we apply this knowledge to study drug treatment, for example? So it's a lot about the similarity between schizophrenia and schizotypy. And I've showed you this image. I promise you, I showed you a picture of a tsunami, even though there's a debate in the art history literature whether this really is a tsunami or just a great wave, but it's called the Great Wave of Kanagawa. It's a picture you probably have seen. And what is the point of this? Well, the point is we're interested in the putative similarity between schizotypy and schizophrenia. And now let's say instead of studying schizophrenia, we study waves, and we sit here in California by the wonderful Pacific Ocean, and we see these small waves rolling against the shore, and we think, oh, that's really interesting. What causes these? What's the etiology of these waves? And we do some research, and we find out, oh, it's the moon, and it's the wind, and that makes these waves. And then a huge big wave comes along, a tsunami, and we think, oh, wow, there must have been a lot of wind, or the moon must have been closer. Right? It's the same sort of causal factors. And of course, that's not the case. So two things that look similar, that differ in scale, differ in magnitude, might have very, very different causes. And that's something we have to bear in mind. That's a limitation of everything I'm going to say now. The brain function, the brain structure that we see in schizotypy, even if it resembles schizophrenia, that may be for reasons that are not known to us. All right, so let's look at brain structure first. I'll show you a couple of studies we did. Veena was involved in this one. Dr. Kumari, I should say, sorry, very informal. And what we did is essentially quite simple. We studied healthy individuals. We even screened them, so there was certainly no one with a DSM diagnosis in this sample. And those individuals underwent structural MRI at 1.5 Tesla. It's a fairly standard structural MRI scan. They also filled in one of the schizotypy questionnaires. Here we used the RISC, that is one of the questionnaires that gives very nice normal distributions. Some of the questionnaires in this field tend to give quite skewed distributions, because most people then say no, and maybe they don't admit to it. And John Rust made this questionnaire to really tap the subtle differences between people, and he tried to really get people to say, yeah, maybe, so that we don't end up with a skewed distribution. And if you look at the question, they're really nicely formulated. So they don't ask, do you hallucinate? But they ask, sometimes my thoughts seem so loud as I can almost hear them. Sometimes seem and almost. And then people begin to say, yeah, actually, sometimes yes. Not always, but sometimes yes. And then you get nice variance between in your data. Okay, so that's the questionnaire. It measures only positive schizotypy, I should say. And the statistics is just to make the point that this is a sample with relatively low scores. So we didn't really have the extremely high scorers in there. The sample had a relatively low mean, but the distribution was very nice, and the reliability, the Cronbach's alpha, was very good. And we correlated this score with gray matter volume across the entire brain with without any search restrictions. We just asked the question, where in the brain are the correlates of positive schizotype? And it turns out they're in two clusters in and around the frontal lobe. The first cluster is medially, that is this cluster here, begins here. It's medially, including the superior and the orbital medial frontal gyrus, gyrus rectus and anterior cingulate. And the second, non-overlapping cluster is laterally. It's in the left hemisphere. It was a trend level there in the right hemisphere as well, but it's significant only in the left, including the insula here, the superior temporal gyrus. This is the inferior frontal gyrus, and this is the middle and superior temporal gyrus. Okay, so it's a frontotemporal cluster. And this is another way of showing the data. This is the SPM glass brain, so it's a see-through brain. The side view, the lateral view, the front or coronal view, and the top-down or horizontal view. And you can see those two clusters. They don't overlap. And what you can also see is there are no other clusters. So it's really a very specific effect. And we also looked at global gray matter volume. So that's the first question. Well, maybe schizotypy is associated with the brain volume overall. No, it isn't. It's literally just in those two clusters. And there were no positive correlations. So this is a negative correlation. This means higher schizotypy scores, less gray matter volume. There were no positive scores. Now, what to make of this finding? Well, if we're interested in relating this to schizophrenia, we need to look into the MRI, structural MRI literature on schizophrenia. And there was a meta-analysis at around the same time on studies using a very similar method of structural MRI images in schizophrenia. And what that study found was that a number of regions are reduced in volume in schizophrenia. But if we look at the ones that are most strongly reduced, so this is the percent of studies reporting deficits, reductions in schizophrenia patients. These include the regions that we found: the left insula, the left inferior frontal gyrus, the left superior temporal, and the left the left superior temporal gyrus. So there are other areas as well. Okay, the left medial temporal lobe, we didn't see that. Parahippocampal areas, we didn't see those. But the areas that were reduced in relation to schizotypy are amongst the most consistently reduced areas of reduction in schizophrenia. Okay, so this would be very similar to the cluster that I just showed you. So that really confirms or provides some evidence for the idea that schizotypy looks a little bit like schizophrenia, not for some fluke, chance reason, but because there are similar brain structural correlates. Okay, so this is what we find. So our schizotypy study agrees with what's been reported in schizophrenia, and since then, there have been a couple of studies that found similar, not exactly the same pattern, but similar findings, especially the left insular finding has been replicated a couple of times, which is nice. There are differences as well. In schizophrenia, there are a lot of areas that are consistently reduced, and we didn't see the thalamus, for example, or the medial temporal lobe, including the hippocampus. They're reduced in schizophrenia quite reliably, but we didn't see those. And then there are other studies showing increases in volume in schizotypy. We didn't see that either. Okay, so it's an individual study. It didn't agree with everything others have found, but there was some good overlap. So after brain structure, let's look at cognition. I know you're interested in cognition as well, and measuring and assessing neuropsychological tests and cognitive function in patients. And cognition is, of course, a vast field, and there are a number of different functions that cognition serves: memory, attention, perception. It's all, broadly speaking, part of cognition: decision-making, reasoning, and so on and so forth. And so we have to necessarily focus on one small aspect of it, and we focus on inhibition. Response inhibition is this, right? Don't do this, don't say this, don't push the button. Terrible things might happen. That's inhibition. Technically, it's any mechanism that reduces or dampens neuronal, mental, or behavioral activity. Okay? And there's a huge debate in the cognitive neuroscience literature about what inhibition actually is, what are the mechanisms, what can be inhibited, how is it inhibited, and so on and so forth. But for our purpose, we're looking at response inhibition, so the ability to not perform a response that is dominant. That means it's automatic, it's easy to do, but in this particular instance, it's inappropriate. I mustn't do it now. like stopping oneself when about to cross the road. You're thinking about something else, and you're stepping into the road, and then you notice the car's coming. You have to stop yourself. It's an important thing to be able to do. Or we've all sometimes said things we shouldn't have said. Oops, just did. Or we sometimes look at things we shouldn't be looking at, right? So we need to inhibit our responses sometimes. And we study this using a very simple task. It's called the anti-saccade task. Again, this goes back to the Institute of Psychiatry days. And what you do in this task is basically what Obama didn't do. So you look straight ahead, and as you do, if you do this, you look at this dot, and soon a dot will appear on the right or on the left. Don't look at it, okay? Here we go. Maybe you did, maybe you didn't. You should have looked over there to the right. It's quite difficult. If we did this again and again, many times, on average, on about 20 to 30% of trials, of runs, you will have looked at the dot on the side, even though you're not supposed to, even though you know that you shouldn't, and you look away from it after you've looked there. And sometimes you don't even realize that you did. It's another question. So that's the anti-saccade task. It's one measure of inhibition, the ability to inhibit reactions that we shouldn't perform. This is what it looks like in the brain. The point of this image really is to show that it's a complex task. It's not a low-level motor task. We see activation in the dorsal cortical network, including the frontal eye field, so the front of the brain, back, frontal lobe, parietal lobe. And these are areas that are also implicated in attention, in working memory, in the volitional control of action. So it's a relatively complex cognitive task. What about anti-saccades and schizophrenia? So now let's turn this around. Let's ask first, has this been studied in schizophrenia? The answer is yes, many times. And what do the results look like? Well, I'll show you our own results from a study that we did in Iceland, where we had access to a very large population or sample of schizophrenia patients and controls. And this is really the typical pattern. What you measure is the percentage of errors. And an error is when you look at the dot when you're not supposed to. And this happens on average about 60% of times in the patient group, and only at about 25-30% of times in the controls. And this is a highly significant difference. If you know about effect size or care about them, this is a large effect size, and it's a very replicable finding. I don't know of a single study out of probably 100 that have not found this. And then in relation to schizotypy, we find a similar effect. Now here the data look very different because we study individuals and we just put them in one group and correlate schizotypy with anti-saccades. And what we see is this. It looks a bit messy, and it is. It's a small effect, but it's there. Positive, higher schizotypy scores go with more anti-saccade errors. So that's a positive correlation. And up here, maybe if you extend this, there will be the highly schizotypal individuals. And they on average, tend to make more errors than the controls and the lower schizotypal individuals. Okay, so in statistical terms, it's an effect that is significant, but it's not a very large effect, explaining 6% of the variance. But that is how large these effects are. We don't get them any bigger, and there are a number of reasons, of course, why this may be the case. But it replicates, or it extends the schizophrenia finding. The next thing we were then interested in, again work done with Wiener, is what does schizotypy look like in the brain in action? So I've shown you the brain structural correlates. Now what if we engage the brain in some activity and correlate that with schizotypy? Because most of what we measure with the schizotypy questionnaire is reflection on behavior, on experiences, so products of the active brain. And so we engage the brain. How do we do this? Well, we could do a number of different things. We can do resting state, fMRI, and we've recently done this actually. You just ask people to lie in the scanner, not do much, keep their eyes open, don't fall asleep, and to just have their own thoughts. You can do this. We've done that, and again, we see similar patterns as in schizophrenia. Or you engage the brain. You give people a task to do. And we gave people the anti-saccade task to do. So this is the question, and we use the anti-saccade task as a behavioral probe, as a means to stimulate the brains. Why? Well, because we know already what the task, what brain areas are active when people perform the task. So we're not in like new territory. We know what to expect. The task is highly reliable. It's a really solid measure. We know what the impairments look like in schizophrenia, and we know what the correlation looks like with schizotypy. So it's a pretty good method to activate the brain and see what are the correlates of brain function with schizotypy. And we did this and find, first of all, at the behavioral level, so we record eye movements and relate it to brain function, related to schizotypy. This is anti-saccade performance in the MRI scanner, and it correlates with positive schizotypy, just like in the other study. The correlation is virtually the same, 0.25 versus 0.26 or something like that. So that's a very nice replication in an independent sample using a slightly different task in terms of the methods, what the task looked like, but the same questionnaire. So it's always nice to have replication. And then while people did this task, we recorded brain activation with an EPI sequence in fMRI. And the correlates of schizotypy in the brain look like this. It's small images, but the point really is we see correlates of schizotypy with brain function in the putamen, in the striatum, in the thalamus, in the cerebellum, and in the visual cortex. And all of these correlations are negative. That means higher schizotypy scores are associated with less brain function, with lower activity. Now, what does this mean? Is this a schizotypy-specific effect, or can we again relate this to schizophrenia? And the answer is yes. There are studies of brain function during antisaccades in schizophrenia, in both patients as well as the siblings, and there is some overlap. The pattern is different, but it's not the same, but there's some overlap. For example, patients also show reduced striatal activation compared to controls. And this is also seen here. So the siblings as well. So striatal hypoactivation, reduced activation in the striatum, seems to be a marker not only of schizophrenia and the siblings, but also schizotypy. And the reduced activation, the lower activation in visual cortex that we saw has also been found before in patients and their relatives, here in a study from Jennifer McDowell's group. Okay? Now, in the patients and the relatives, you can see a lot of other activations or differences. We didn't see those. So schizotypy is not the same as schizophrenia. Of course it isn't. We wouldn't argue it is, but there's some overlap. So let's summarize what I've said so far. We observe these inhibition deficits in schizotypy using an anti-saccade task. We've seen it a couple of times. I'll show you another study where you see it again, so it seems to be a replicable effect. Higher level of schizotypy is associated with reduced bold signal, a measure of brain activation. And overall, this suggests that there are some similarities between schizotypy and schizophrenia patients, as well as their relatives, in terms of behavior and brain function. And what I haven't said is that the relatives also show anti-saccade impairments on a number of different measures. So schizotypy really looks a little bit like schizophrenia at the symptomatic, at the phenotypic level, but also in terms of the brain structure and brain function. But schizotypy does not mirror all of the characteristics of schizophrenia. We wouldn't expect it to. It is something different. I'm talking here about healthy individuals. So what is the point then? It's been interesting to find this, and like I said, we can try and relate this to or use this to understand the etiology of schizophrenia. But perhaps we can also use this knowledge for some other more applied aspect of schizophrenia research. So perhaps schizotypy can serve as a model system. And we work a lot with model systems, of course, especially in drug development. So schizotypy could be a subclinical model system of schizophrenia. What are models? Especially in drug development studies, new compounds are at the preclinical level in animals and at the clinical or human level often tested before they're administered to patients, tried on patients, administered in model systems. And what's a model? Well, a model, like a toy train, it mimics the real thing. It's not the real thing, but it's easier to handle. You can move it and see how it behaves and then apply this to the real thing and see maybe that's how my intervention behaves in the real world as well. People work with animal models, and of course there's some expertise in animal models here as well, like isolation rearing. That's a model, is thought to be a model of schizophrenia, or PCP administration, ketamine, and then there are human models as well. We have to be a lot more, it's a lot more difficult to do human models, but ketamine administration may be a model. It's a pretty good model of psychosis. Sleep deprivation, which is what we've been studying over the last years, 24-hour sleep deprivation is mimics, models some of the effects of schizophrenia. So there's a lot of work using these models, and here we tried to investigate whether schizotypy could be another one of these models, a trait, not a state model. Giving ketamine or sleep depriving someone for a day or a day and a half is a state model. We'd expect to anything we induce in the brains of these people to fully reverse, to fully go away when we switch off the ketamine pump or they get a rest, a good night's sleep. Okay, these are state models, but trait models are maybe like isolation rearing. Probably once these animals have been reared like that, they have lasting effects. You'd know better than I, but I would assume it's a long-lasting effect. And schizotypy probably as well, because we know schizotypy is a set of traits. The scores don't go up and down from one day to the next. They're highly reliable and they show developmental trajectories, of course, but they're relatively stable. So why would we use schizotypy? Well, it looks a little bit like schizophrenia, but these individuals, people with high scores, are not patients, so they are not usually treated. They don't have actual symptoms that may confound cognitive assessments. Subjects are relatively readily available. By that I mean we have to screen and we have to go through a lot of questionnaires to get high scores, but they're there to be tested. And so potentially we can then use this to detect immediate drug effects on state markers or on biomarkers. So the questions we pursued in this study are, can we use the anti-CK task as a biomarker, as a marker of a drug effect in high schizotypy, in a high schizotypy model? And what are the drugs whose effects we want to predict? Well, schizophrenia-related drugs, like antipsychotics, risperidone and anisulpride, or cognitive enhancers, like nicotine. There's a very large literature on nicotine and schizophrenia, and Dr. Kumari has contributed a lot to it by arguing, proposing the self-medication hypothesis. One of the reasons people with schizophrenia will smoke more frequently and more than healthy controls may be to enhance cognition. There may be other reasons, but that is likely to be one of the key reasons. And so that's one of the reasons we were interested in nicotine as a cognitive enhancer, as a substance that improves cognitive function. And so we attempted in this study, and this is really the last study I'm showing you, to validate the schizotypy model with regards to antisaccades as a biomarker. So what did we do? So first of all, what are the hypotheses? We study high and low schizotypes. In a drug study, we can't give drugs to 100 or 200, well, we did, but in a drug study, it's really statistically and methodologically easier if you have separate groups. And then you have group as a factor in your model. So we used high and medium schizotypal subjects. And the first thing we would expect is that the high schizotypes would show increased antisaccade errors relative to the controls. That would be a replication. And we would expect to see this under placebo. The next thing we expected was that the high, but not the medium schizotypes, would benefit from risperidone. Why? Well, risperidone is a clinically effective antipsychotic, and it's been shown to improve antisaccade performance in schizophrenia. So if we now give individuals who indicate in their questionnaires that they experience some schizophrenia-like symptoms, and if they show increased antisaccade error rates, an antipsychotic, risperidone, then really they should get better at antisaccades. The controls, we wouldn't expect to improve. They don't need a drug like that. They might even get worse. Why did we use medium schizotypes? That's a really good question, and we would have probably had certainly had more power if we had used low schizotypes. It was a consortium decision at the time to study the normal control group rather than a very low schizotype group, because we didn't know a priori what are low schizotypes like. Maybe they're statistically unusual by definition, so that's why we went for the normal group. We did in an extension of this. With Ivan Koichev in Manchester, a an add-on where we, in addition to the high and the median schizotypes, included low schizotypes. And in terms of some of the cognitive markers we looked at, like smooth pursuit and antisaccades and NBEC, I think, they were even better. So it's really, the correlation is there, and if we'd sampled from the low end of the distribution, we probably would have gotten stronger effects. So that's what we would expect with regards to risperidone. Amisulpride really is a comparison compound. We wouldn't expect any effects on antisaccades. And then we would expect both groups to benefit from nicotine. Why? Because previous studies have shown that schizophrenia patients get better in antisaccades when they receive nicotine. Relatives, healthy individuals, smokers as well as non-smokers. So nicotine is good for everyone. That's what we expected, and I'll say that's what we found. At some level, yes. Sorry,
Ulrich, how do you know that this is not just an intervening factor? How do we relate this to schizophrenia in the sense that you can have a compound A, in this case it's risperidone, and it is improving. It is improving antisaccades, but the fact that something improves antisaccades, would it also have effect as an antipsychotic? Suppose I give you, for example, we could say that a cognitive enhancer that is used in Alzheimer's might improve cognition. In MCI, mild cognitive impairment patients, does it, and we know that there are cognitive deficits in schizophrenia. So it's the question then is that if a compound reverses or changes a factor or a function in schizotypy, how would that relate to, why would you think of it as an intermediary factor in the illness? I'm not expressing myself that clearly.
I think I know what you're getting at.
My point is why it could be any, that could be an end in itself rather than a means to an end. You see that there is you could say that risperidone changes antisaccades in schizotypy, for example. How do we then make that conceptual leap from that factor onto schizophrenia?
Right. I think, it's a really good question, because it could happen for any reason. But I think the key is really the medium or low schizotypy comparison group. If they don't benefit, then the best bet we have to explain this difference is the relatedness to schizophrenia and the high schizotype. So there is something in their brains, and I've shown you some brain data, that would benefit from risperidone, whereas the controls might not.
Because if you showed this in the prodrome, one would say, okay, because the people that are allegedly in the prodrome, you never know who's actually in prodrome till that. You can only retrospectively make that assumption. But in any case, you can say they are on that continuum, that they're going on to develop psychosis, schizophrenia. Here, I think there's data that you've shown and has been evident for a while that this is not on the continuum. It's on a plane by itself, but shares similar characteristics to schizophrenia. So whether they, I think it's an interesting model. The things that are difficult is how do you translate this into a drug development protocol?
Right.
We've struggled with this when we've done the cognitive enhancers for Alzheimer's. We had studies, as you remember.
Yeah. So it's a difficult question. So this was work done with industry, and they were interested in developing, testing, validating putative models of psychosis. So in a parallel study, we used ketamine and then also administered risperidone. And so their idea was, let's find reliable, practical models that can serve to screen for compounds and with regards to certain biomarkers, so that compounds are put through this test, and if they have an effect, if they get into the brain, if they improve cognition, antisaccades, whatever, they go on. If not, it's a stop. That was the dream in a way. And
marker or a surrogate marker in terms of saying, okay, this might be.
You can say
it could be a surrogate marker.
it would or it should, at least in theory, translate given the overlap at the brain structure and function level, and the fact that yes, most of them will not convert to being in frank psychotic.
Yeah.
They still have higher chances than the general population.
I mean,
Yeah. Has kind of, for a long time, tried to translate the animal models, which we see results very clearly in the animal models, and try and translate that into what are the putative human models, if you like, for surrogate markers. And I think this is a very interesting idea. The question is, what do we do in terms of making it happen?
Yeah, well, that's a big question.
But your results will show you. I mean, I stopped you before you got your results, but the fact that it's an antipsychotic and it's used all the time with good effect.
Yeah.
So tell us what it's like.
I should have maybe included, but like a slide on limitations. But we can just I do this sort of impromptu because there are clearly limitations of this. I mean, there is no animal equivalent of this, right? Maybe isolation rearing, but having no friends on Facebook isn't the same as being reared alone in a cage. So that's one limitation to begin with. But let's look at this in a minute. So what we then did was to actually test a very large sample because it was a between subjects design. And so it's parallel groups, double-blind, randomized, placebo-controlled, over 200 subjects. And those 233 subjects are first, you have to split them into high and medium groups, okay? So about half of them had high schizotypy scores and the other half had medium schizotypy scores, putatively normal scores. And then each of these two groups goes into one of four drug groups: 2 milligram low dose risperidone, 400 milligrams amisulpride, 7 milligram nicotine patch, and or placebo. So each individual does only one of these things, is either high or low schizotypal and gets one of those three drugs or a placebo, or between subjects. That's what it looks like. So if you to get an idea of the sample sizes, round about 30 in each of the combinations: high schizotypal risperidone, low schizotypal risperidone, and so on and so forth. And they didn't just perform antisaccades on drug. They performed a cognitive battery, including verbal fluency and NBEC, and I mentioned those results again in a minute, or those tasks, because they showed quite good results as well. So looking at the antisaccades, this is the key result. Again, on the vertical axis are the direction errors: higher scores, worse performance. Here the medium, the controls, medium schizotypes. Here the highest schizotypes. So the first thing we were interested in, under placebo, if we just take out the placebo groups, do they differ? And the answer is they do. That's this line here. The high schizotypes on placebo make more errors than the low schizotypes. So this is the third study I'm showing you, and the third study we've done that finds this effect. But then when we challenge them, this happens. There's a main effect of drug, which isn't particularly meaningful because there are four different groups, but also a drug-by-group interaction. And this is a lot more interesting. We were expecting interactive effects. The interactive effect, that's a soundtrack to it, I guess.
Well, I guess a lot of hot topics.
Exactly, inside as well as outside. So the interaction really suggests that, so if you resolve this into smaller models, the first says there's a main effect of nicotine. So across groups, if you collapse these and you compare nicotine with placebo, then error rates are lower under nicotine than under placebo. And it looks like this effect is stronger in the high schizotypes, but statistically it isn't. So irrespective of schizotypy, as expected, everyone does get a bit better, or people are a bit better on nicotine than on placebo. And then the interaction comes from antipsychotics compared to placebo. And this effect suggests that this effect depends on group, and this suggests that in the medium schizotypes, if you look what happens when you give risperidone, the error rates go up. They are higher than when you give placebo. So in the normal control group, if you like, as expected, risperidone isn't good. The error rates are higher than with placebo. In the high schizotype group, the effect, there's no significant difference. If anything, it's numerically in the opposite direction, but it's not significant. So, did this work? Well, in part. What we found was that schizotypy predicts the response to the drugs, to risperidone, but not in the way we expected, or at least only partially in the way we expected. This we might have expected. If you don't need an antipsychotic, it's not good for you. Here, we would have expected this to be significant. And in terms of some of the other tasks that we looked at, like verbal fluency and NBEC, that was the case. So those other tasks were actually better in detecting positive drug effects, antipsychotic effects in the high schizotypes. So let's summarize this. We replicate the main finding, increased errors in schizotype and the placebo. Then risperidone impairs performance in controls and has no detectable effect in the schizotypals, even though there may have been a tendency. Overall, the level of schizotypy does predict the cognitive, the brain functional response to antipsychotics. And the interpretation of this is now, it's now difficult. So we set out really to model this, to test this model of schizotypy. And if I was a drug company and I saw this, I would say, well, it's interesting, but it's probably not the model we want. Maybe ketamine is a better model, maybe amphetamine is a better model, selective of only the positive symptoms, but maybe it's a stronger model. Because what we really expected that with regards to this biomarker, the high schizotypes get better, we didn't really see. With regards to other tasks, yes, but here we didn't see it. To me, it's still a very, very interesting finding because I'm primarily interested in understanding schizotypy. And here's a quote from William Carpenter, which is really interesting in this context. And he asks, what is the most defining feature of persons with a diagnosis of schizophrenia? And he says, the answer, it's not some complex collection of symptoms, the answer is the incredible tolerance to dopamine antagonists. And that is exactly what we see. Healthy people with medium schizotypy scores don't tolerate an antipsychotics in terms of their performance, they get worse. But the high schizotypals at least tolerated the drug. Okay, so let me summarize then. Schizotypal personality looks a little bit like the symptoms of schizophrenia. Schizotypy co-varies, is correlated with brain structure, brain function. It predicts the response to an antipsychotic. Overall, we think this is evidence of a continuum from normal experience, normal personality to clinical impairment. And the implications are, I think, manifold. It's important to study this further in order to better understand schizotypy in its own right, because it's not generally a positive phenotype, a trait to have or set of traits to have. Then we think, I think we can learn about the etiology and conceptualization of schizophrenia. What is schizophrenia? What are the fringes of schizophrenia? Where do we draw the line? And possibly we could use this idea or this model system for drug development, although I'm really quite cautious and I won't make a strong claim about this with regards to the anti-psychiatry task. So let me thank everyone who's done this work. Veena has been really instrumental in virtually all of these studies, I think, if not most, but probably all of them.
Sure.
And then my team at the University of Bonn, of course. And thank you for your attention. Happy to take questions. Yeah.
So I have some questions for the brain structure and schizotypy. You presented results showing the clusters. Those were all cortical. Did you look subcortically and not find anything?
We looked as well.
Okay, so this is
We just didn't find it. Yeah.
Okay.
Yeah.
I think if you combine that with the functional findings, we do find subcortical abnormalities when you go to the function level.
Which was going to be my next thing, which was in your antipsychotic task.
Yeah.
You didn't see any relationship with the orbitofrontal cortex?
No, we didn't see any correlation with it.
Very pleased.
Why are you pleased?
I'm big on the
Because he likes
OFC not being involved in response inhibition at all training.
Okay. What about emotional response inhibition? That's direct projections to the amygdala, right?
I believe in Jeffrey Schoenbaum's results showing that it's involved in inferred value rather than in direct response inhibition. So it's a little more complicated, but I really like your result, and I love this task, this antipsychotic.
Okay.
It's very elegant.
Thank you.
And then the you found a negative correlation in the fMRI between the putamen and the task?
And the performance on the task, yeah.
All right.
Yeah.
Cool. I just wanted to make sure I got that right.
Yeah.
I think there is some evidence, and I may not be putting it correctly in siblings, that if the task is very difficult, you might even get a hyper response.
Right, so the question is, what does task difficulty do to differences? And I don't know. The answer is I don't know. But if you vary the difficulty of the task, then in the healthy brain with increasing task difficulty, you tend to see an increase, and maybe at some point people just stop doing it. If they can't, then the signal would go down. And in the patients or relatives, the curve may be shifted. But we haven't done this. Yeah.
And I guess there might be similar issues when you see poor performance in low to medium schizotypes under antipsychotics. So there might be too little dopamine. Just like too much dopamine is bad for performance, too little dopamine may also be bad.
In the high schizotypes?
Yes, so in your medium schizotypes, if they are kind of optimized, if you give antipsychotics, and dopamine goes too low.
Yeah, yes.
So there could be an inverted U, and I think we need to understand relationships at every level of the curve.
Yeah,
to actually figure out whether this model is going to be of any use.
Right, that's a very large study I've always wanted to do. I mean, maybe, Dr. Sharma would be interested in funding the study to be run here. So we need multiple doses of drugs, people who systematically differ in the baseline dopamine, and parametric task variations in task difficulty. And it becomes a very, very large study with lots of cells. People differ by baseline dopamine, whichever way you measure it. People differ by they get different drugs.
Maybe we could perhaps make it more direct. So schizotypy is self-report. What about just simply using blink rate as the model?
That could be a very simple way of stratifying subjects into high and low striatal dopamine activity. So striatal dopamine is associated with blink rate. The more you blink, higher your striatal dopamine. And you can't, well, that's the link. And so that could be a very simple way of screening subjects into high and low, or high, medium, and low. And then each of these gets probably 3 different doses of drugs and a placebo, and does 3 or 4 different variants of the task. Yeah, but that's a study I'd like to do at some point.
So there's work showing that modafinil increases response inhibition in methamphetamine users, but not in healthy controls.
Right.
Is there, that, which reminds me a little bit of your data, is there any work with modafinil on this task in schizotypy?
Oh, that's not in that combination. I'm not sure whether there are even any studies of modafinil on antisaccades. So we've studied methylphenidate and atomoxetine and their effects on inhibition in just healthy but very carefully screened healthy controls, and we don't find behavioral improvements. We see effects in the brain. I mean, the drugs get there. Methylphenidate changes bold in the striatum where the dopamine transporter is, which is blocks. Atomoxetine has really strong effects in the medial frontal cortex. So the drugs get into the brain, they change the activity, but it doesn't translate into a behavioral improvement. So we think maybe our tasks aren't difficult enough. So we haven't seen really positive, beneficial effects of methylphenidate or atomoxetine on inhibition. Yes.
Just one question. I want to make sure that I understood. So in response to gray matter reduction, did you state that in high schizotypy they actually had an increase in gray matter?
No, they have less gray matter. In the areas that I showed you. There's one other finding that's come up a couple of times, and this is a complex literature because there are different questionnaires, and the construct, schizotypy itself is sort of a well, it's a complex construct, and then imaging allows a lot of different approaches to data analysis. So it's a complex literature, and if you see a finding come up twice or several times, then that's likely to be a very strong signal. And so one other finding that has come up a couple of times is actually increased gray matter volume in relation to schizotypy, but in the posterior medial parietal cortex, in the precuneus posterior cingulate. A couple of, I think 2 or 3 groups have found this. We didn't see it. We saw less gray matter volume in frontal temporal areas in high schizotypy. Yeah.
It might be that the behavior is only when you really have extreme schizotypy scores. Even in schizophrenia, I think when it comes to precuneus and various findings, sometimes you do have
Okay.
Very interesting.
Yeah.
Okay.
What does the genetic studies stage remind me
Of schizotypy? So far, not much. There have been a few individual sort of candidate gene association studies, but I think you can.
Nothing obviously it's too rare for there to be twin studies.
Oh no, they're twin studies. Heritability is good. We've published twin data on this. Heritability is, as you'd expect it, 40-50%. That's what you get with personality traits. Heritability of anti-saccades, we found this as well, it was about 50%. Now, the really interesting thing though is, and it's not out yet, a colleague of mine told me that they did a GWAS of schizotypy. No, sorry, they used the polygenic risk score derived from a GWAS of schizophrenia and related that to schizotypy. Now, after all this, we would expect that schizotypy correlates positively with the polygenic risk score. Polygenic risk score is adding all the variants that are related to schizophrenia and sort of counting the number of risk variants you have. And the more you have of these, the closer you are to schizophrenia, right? So you'd have higher scores, and they found the opposite relationship. And that really would be, if it's true, something of a paradigm shift, because then I'd have to say, well, the link is there, but genetically they go in opposite directions. But it's not published yet, I haven't seen it.
I don't think it's necessarily as contrarian as you're stating, because if we assume that it's not on the pathway to schizophrenia, but it is there.
Sorry, it's not on the temporal pathway, yes. You mean? No, yeah, I agree.
And that finding will actually make sense
right.
rather than being contrarian.
Right, I actually agree.
That says that this is not a trajectory that will take you to schizophrenia. This is a separate entity and exists on its own separate plane, if you like.
Yeah.
Sharing some traits with schizophrenia.
I
have a mother who's texting me or contacting me on LinkedIn from Florida to help her son, and she's got partial expression of the gene, I'm sure.
Right.
She goes off, goes cold, and now thinks that I'm the best things in sliced bread, saying, can you help my son? Nobody else picks it, which is the type of patient that often accepts. The point is this though, how does that kind of finding helps us understand that, and helps us use schizotypy as a much better example for testing of surrogate markers, because it's a stable entity and it's not fluctuating. It exists on a different plane. You're checking all those boxes that are required, because if you give ketamine to humans, I've had them be really psychotic with ketamine, and then this is a transient phenomena, it goes away. Here you've got a stable trait. If you can make a difference in a stable trait, and your finding that you just mentioned from your colleague would give further credence to the fact that this is on a different plane.
Well, it
exists and makes a good marker.
There are two possible, at least two possible ways this might go. One is schizotypy is everything of schizophrenia but less. That's possible. So that there would be a correlation then with the genetic risk score, but just not as high. And then obviously you won't get those who have all of those 104 SNPs that have been associated, because they'd be in the schizophrenia category. So that's one possible scenario. But then I'm not sure how likely that is, etiologically speaking, that everything that's associated with schizophrenia is there at a lesser level in schizotypy. Another model would be that some of it is there at a lesser level, maybe some even at quite a high level, maybe some people really have a hallucination, but then there's some protective factor, and that goes the opposite way. And that's why they don't get the diagnosis. And we're currently doing this. We're at a very different, so it's really interesting. Where are these protective
factors? Sorry, this is a partial expression, almost. You were going to develop schizophrenia, but because of the protective factors, you're not developing schizophrenia. Then it's the partial expression hypothesis, where you're saying, okay, they were on the path, but then something happened that got them away from that path. There
Possibly.
would be another one which says that they were not on the path to schizophrenia, and that these are traits that they have. We know that personality traits arise in adolescence, they remain throughout the year. It's very difficult to change any of that. So what do we do in that case? That some of these traits are expressed in a much to the much greater intensity in schizophrenia. But it's like having personality traits, it's like having conduct disorder, which we see with the kids in our San Diego facility. And that translating into antisocial, full-blown antisocial personality disorder, then it's very, very difficult to treat access to. And so I don't have, it's been a long time since we did research, but my thoughts would be that we conceptualize this as a stable trait rather than it was on the pathway and then got stopped. But I've not read the literature on this recently. But if it's a stable trait, then you have a much better and on a separate plane and not on a trajectory. Then you have a much better substrate, if you like, for it to be a condition in which you test surrogate markers. Because then it's a stable trait and not something that's evanescent, like the ketamine models that John, people at Yale have been using for a while. But anyway, there's and the fact that it's heritable, it fulfills all the other criteria of a personality issue, but then you've got the expression rather than a partial expression. I think the partial expressions are there in the siblings, the parents, and that they've had protective factor. When we did this multi-family studies, including imaging, you saw a lot of people that were kind of quotes unquote, people would call them odd, I guess, but there's nothing that you can put your finger on. And they didn't develop this. They get a highly scientific way of describing people as odd. Anyway.
It's human pattern recognition. That is human pattern recognition, to call someone odd. So there's a lot of work now in MRI statistics using supported vector machines trying to detect patterns in MRI images to predict who converts and who doesn't. But we just do this, we look at someone and say, oh, they're odd, and we don't know why, but everything we know, everything we perceive flows into this judgment. So it's layman's lay speak, but it's, well, it's not a word to use, but of course I know exactly what you mean, and we'll do. Yeah, Vina.
Another interesting way would be to look at state-trait interactions. So you have people who have by trait induce some states, such as sleep deprived.
Well, we did that with sleep deprivation, right? So we sleep deprived high schizotypes, and in some domains they respond then even more strongly than low schizotypes. So.
Very good.
Good, thank you
All right.
very much.
Thank you.